Hello, I am Professor Sims, and in this video I will discuss biochemical differential tests, specifically those related to fermentation, cellular respiration, and reduction potential. This is the 7th in the series of 10 lab sessions held as part of my Laboratory for the Fundamentals of Microbiology course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, and due dates, and all of your other course information. The learning objectives for this unit include observing and describing the process and byproducts of fermentation of simple sugars, becoming familiar with the concept of an organism's reduction potential, the role of coenzymes and oxidation, and learning about aerobic, anaerobic respiration, and as always, understanding safety and disposal procedures related to these experiments. So let's look at metabolic pathways. Every living organism carries out some form of glycolysis, suggesting that this mechanism is an ancient universal metabolic process. The process itself does not use oxygen. However, glycolysis can be coupled with additional metabolic processes that are either aerobic or anaerobic. So glycolysis begins with a single 6-carbon glucose molecule, and it ends with 2 molecules of a 3-carbon sugar called pyruvate. Pyruvate may be broken down further after glycolysis to harness more energy through aerobic or anaerobic respiration, but for many microorganisms, including a lot of the bacteria we work with, a lot of them are unable to do the cellular respiration. So for these guys, glycolysis is their only way of generating energy in the form of ATP. Next, you have the Krebs cycle. This is named after its discoverer, Hans Krebs, and it's also called the citric acid cycle. You may have heard of that. Unlike glycolysis, the Krebs cycle is a closed loop. So the last part of the pathway regenerates the compound that is used in the first step. So that's why it's called a cycle. As one turn of the cycle returns to the starting point, the cycle produces 2 CO2 molecules, 1 ATP molecule, 3 molecules of NADH, and 1 molecule of FADH2. And Krebs occurs in both aerobic and anaerobic respiration. Then you have the ETC, the electron transport chain. The electron transport chain comprises a series of membrane-associated protein complexes and associated mobile accessory electron carriers. So these electron carriers pass electrons along in the ETC, and it all happens because of their redox potential. So for a protein or a chemical to accept electrons, it must have a more positive redox potential than the electron donor does. So electrons move from electron carriers with a more negative redox to those with a more positive redox. So it's all happening on a proton gradient, and it is producing a large amount of ATP, and it does require oxygen. So this is only happening in aerobic respiration. Speaking of aerobic respiration, aerobic respiration uses oxygen to produce energy in the form of ATP. Byproducts of aerobic respiration include carbon dioxide and hydrogen peroxide. Usually, those are the big ones. Anaerobic respiration uses nitrate, sulfate, or another highly oxidized inorganic substance to produce ATP. So it uses those instead of oxygen. Byproducts of anaerobic respiration can include things like ethyl alcohol, hydrogen gas, and carbon dioxide. One type of anaerobic respiration that occurs in a lot of the bacteria that we have heard about is carbohydrate fermentation. Carbohydrate fermentation is the anaerobic oxidation of sugars to produce NADH, which is then converted to ATP via glycolysis. Byproducts of carbohydrate fermentation include your organic acids, and these lower the pH of your growth media. And also gases like H2, CO2. These can be observed if they can be captured, and more on that in a minute. I'll show you in just a minute. Byproducts of fermentation that we use in the industry include lactic acid. We use that to make yogurt and cottage cheese and sauerkraut. Ethyl alcohol is a byproduct of yeast fermentation, and we use that to produce beer and wine. You know, that's the alcohol that you can drink. And then carbon dioxide which is also a byproduct of yeast fermentation, is actually used to make bread. It makes the bread rise. We're going to do the carbohydrate fermentation test, and I just want to kind of explain the phenol red fermentation assemblies that we'll be using. This includes a nutrient broth that contains a carbohydrate, so each broth is going to have either lactose, sucrose, or glucose sugar in it. And it also includes let me back up and explain to y'all that when I say sugar and carbohydrate interchangeably, a carbohydrate and a sugar differ based on the amount of monomers that they have, so it's based on size. So the smallest one is going to be a sugar. In fact, the smallest sugar is glucose. And then your larger sugars are polysaccharides or carbohydrates. And then even larger than that would be your starches. Okay, so don't let me confuse you if I say sugar instead of carbohydrate and vice versa. They're essentially the same thing. You have a nutrient broth containing a carbohydrate and a pH indicator. The pH indicator is phenol red. And then you have a small inverted test tube in the broth. This is called a Durham tube, and that is used to capture any gas formation, so your hydrogen or CO2 gas. We're going to determine whether or not bacteria can ferment the lactose, sucrose, or glucose, whether or not the bacteria can actually survive in anaerobic conditions, right? So anaerobic, respiration, fermentation, these things are hand in hand. And you may end up utilizing this test to help identify your unknown specimen as well. So for experiment 1, for each organism that you test, you're going to need 3 tubes. One tube is going to serve as a negative control, and you're going to use that negative control to compare the colors. So after they incubate, you may have something that is actually positive for acid production, but it hasn't turned all the way yellow. It's actually kind of more of an orangish color. And you may have observed that tube and you're saying that it's a negative result, but that would be a false negative. So what we do is we use a control that has not been incubated, or I'm sorry, it has been incubated, but it's not been inoculated. There's no bacteria in it. And you hold that up next to your test tube and see if the color has turned more yellow or if it stayed the same. And that helps you to determine if you have acid production. And we'll talk about this some more in lab. So what you're going to do is you're going to use your sterile inoculating loop. You're going to inoculate each of the test tubes, but not the negative control tube. And after the tubes are incubated, you're going to score them for either the production of acid or the production of gas. If you have gas, it should be a bubble in your Durham tube. And for this example, so let's just go through this one. The tube on the left demonstrates a negative fermentation reaction. The broth has a high pH. There's no gas in the Durham tube. Okay. The tube in the middle, this is actually showing fermentation. This is a positive fermentation test, and it's positive because of the color change. It's going from a red to a yellow. That indicates that the pH has dropped, and that is due to acid being produced. And then the tube on the right is also positive for fermentation, and it is actually showing production of acid and the production of gas. So it's illustrating fermentation via both indicators in the test tube on the right. The next test that we're going to do is the Catalase Test. Catalase breaks down toxic hydrogen peroxide into harmless water and oxygen. This test is often used to identify bacteria of the Staphylococcus genus. If you do a gram stain on a bacteria and you have cocci and you're not sure if it's Streptococcus or Staphylococcus, you can do a Catalase test, and that will determine which one you have. Staphylococcus should be positive. Streptococcus should always be negative. Bacteria that produce catalase can be detected easily using typical store-bought hydrogen peroxide. This test can be performed on a microscope slide, which is how we're going to do it in lab, but you can also do it just by adding hydrogen peroxide directly to the medium containing bacterial growth. These figures both show hydrogen peroxide reacting with the enzyme catalase. It is quickly broken down into water and oxygen, resulting in the bubbling. This tube on the right down here is showing a negative result. That is, this bacteria does not produce catalase here, but the other two do. In fact, If you put, this is why you sometimes don't want to put the hydrogen peroxide directly on your specimen, because these guys over here that are not producing catalase, they're actually being killed by the hydrogen peroxide. For experiment 2, you're going to do it on a slide. You're going to obtain a clean slide. If you have fingerprints or smudges, you clean it with lens paper. You're going to put your organism directly on the slide using aseptic technique, and then you're going to add like 3 or 4 drops of hydrogen peroxide right on top of your specimen, and then you're going to observe it for bubbling, which would indicate a positive result. So this reaction can be more or less vigorous depending on how many or how few cells that you have on your slide, but it should happen immediately. So if you have any bubbles form, then that is positive for catalase. That means that that reaction has occurred. In aerobic respiration, the final electron acceptor at the end of the electron transport chain is an oxygen molecule that becomes reduced to water by the final electron transport chain carrier, which is cytochrome oxidase. Cytochrome oxidase differs between bacterial types, different species, and it can be used to differentiate closely related bacteria, and it's used often in diagnoses. So at the very least, if a specimen produces cytochrome C oxidase, then you know it can tolerate oxygen and use the electron transport chain, so that's good information to have. The gram-negative, so here's an example, the gram-negative opportunistic Pseudomonas aeruginosa and the gram-negative cholera-causing Vibrio cholera. They both use cytochrome C oxidase, which can be detected by the oxidase test, whereas other gram-negative Enterobacteriaceae, like E. coli, are going to be negative for this test because they produce different cytochrome oxidase types. For experiment 3, your instructor is going to provide you with a dry slide, and you're going to place 2 drops of deionized distilled water on the filter paper. This will activate the reagent that's in the paper. You're going to either, if you're taking from a broth culture, you're just going to put your wooden applicator stick into the broth culture. If you're using a plated culture, you're going to scrape a small amount of bacteria off of the plate. And once you have your inoculum, then you're going to put that on the filter paper. You want to make sure that you do not use an inoculating loop for the oxidase test because metal oxidizes and it's going to mess up your results. You apply your specimen to the filter paper, and what you're looking for is for a color change. Positive results for this test should appear in about 20 seconds. Sometimes it takes a little bit longer. When I give these to you, or when your instructor gives these to you, we'll put them in a petri dish so they can kind of sit there without getting contaminated from the outside. Sometimes it takes a little bit longer. It should be about 20 seconds to a minute though. The dark blue color is what you're looking for. So this guy up here on the upper left is positive for oxidase, cytochrome C oxidase. This guy over here on the right is negative, and then these 2 down here have not been inoculated. So have a look through your Lab 7 specimen. Again, the most dangerous one that we're working with here is your Pseudomonas aeruginosa. This guy's pretty nasty. He's an opportunistic pathogen, but he is the bacteria that produces or it causes the blue-green pus. So it can cause a pretty nasty infection if you're not careful. So you want to be careful with him. Lactococcus lactis, he's new. We haven't worked with him before. He is completely non-pathogenic. In fact, he is used to make buttermilk and cheese and other kinds of stuff, yogurt, and he's actually quite beneficial to humans, and he's used to make antibiotics and things for treating other infections. You do want to make sure, as usual, to go through your safety guidelines. We're running a lot of different tests this week, and well, for Lab 7 and Lab 8, so be very mindful of your aseptic technique. Be careful with your bacticinerators and hot plates. So we're actually only running 3 tests in Lab 7, and 2 of them you're going to get your results right away, and they're just yes or no, your catalase and oxidase tests. And then for Lab 7, I'm going to go through it again with you, and it's time to come back and look at them. But the carbohydrate fermentation test, you're going to be looking for acid production and or gas production. Okay, so it's pretty straightforward. I want to thank you guys for watching. Don't forget to do your reading. Check the description of this video for more videos related to these topics, and leave your questions for me in the comments below.